Welcome back, everyone. Today we're doing a deep dive on something pretty fundamental in microbiology, the methyl red test.
Right.
You might not think about it every day, but
Yeah.
this test actually plays a big role in all sorts of things, from figuring out what kind of infection someone has to making sure your yogurt tastes, well, like yogurt.
Absolutely.
Yeah.
Basically, the methyl red test helps us understand how different types of bacteria, those microscopic powerhouses, process their food.
And we're talking specifically about...
Glucose. Primarily glucose.
Okay, so why is that so important? Why do we care how bacteria break down glucose?
Because it helps us tell them apart. You see, just like you might prefer a salty snack and I might want something sweet, different bacteria break down glucose in different ways, producing different byproducts. The methyl red test helps us identify those byproducts and from that, identify the bacteria.
Okay, that makes sense.
Right.
So how does this test actually work? Walk me through it.
All right, picture this. A delicious, bacteria-friendly broth. We call it MRVP broth. It's got everything a growing bacterium could want. Glucose for energy, peptone for protein, and a special ingredient called a buffer.
Wait, a buffer? What's that all about?
It's actually crucial. The buffer helps keep the broth's pH stable at the beginning.
And that matters because...
We want to see if the bacteria themselves change the pH by producing acids as they, do their thing and metabolize. The buffer lets us isolate the effects of the bacteria themselves on the pH.
Ah, I get it. So we add the bacteria to this broth, and then what? We just let them hang out.
Pretty much. We let them grow in the broth for 48 hours.
2 whole days. Why so long?
It gives them plenty of time to break down the glucose, and if they're the type that produce stable acids as a byproduct of that breakdown, to make enough that we can measure it accurately.
Makes sense. So after 48 hours, what's next?
We take a little sample of the bacteria-infused broth and add a few drops of, you could say, our magic ingredient, methyl red reagent.
And this is where things get colorful, right?
You got it.
So what happens?
The methyl red, it acts as a pH indicator. Like a detective, it tells us whether those bacteria have been busy making the broth more acidic.
Okay, so if the bacteria are producing acid, what do we see? What does the methyl red tell us? So if the bacteria are producing acid, what do we see? What does the methyl red tell us?
If the bacteria are producing a lot of stable acids, the broth will turn a bright red color.
Okay.
It means the bacteria fermented the glucose in a way that made the broth significantly more acidic. Technically, it drops the pH to below 4.4.
Wow. So it's like the bacteria threw a little acid party in the broth?
Yeah, something like that. But what if we add the methyl red and the broth stays pretty much the same color?
What does that mean?
Well, if it stays yellow, it means a negative result.
Yeah.
The bacteria didn't produce enough stable acids to really change the pH of the broth, so it's probably still above 6.2 or so.
So it doesn't mean the bacteria aren't doing anything, right? They're
Okay.
just...
Right. They might still be fermenting the glucose, but they're producing different byproducts. They're using a different metabolic pathway.
It's like they're playing a different metabolic tune.
Precisely. And this is why the methyl red test is so valuable. It lets us differentiate between types of bacteria based on what their metabolism produces, kind of like how we can tell people apart by their fingerprints.
Fascinating. So we're not just looking at what they eat, but how they digest, so to
Exactly.
speak.
Yeah.
Can you give me an example? Like, what are some of these bacteria and how do their acid-producing tendencies vary?
Sure. Let's talk about Escherichia coli. You probably know it better as E. coli.
Oh, yeah, E. coli. Everyone's heard of E. coli.
Right. So E. coli, which is commonly found in the intestines of humans and animals, well, E. coli would give you a bright red methyl red test,
Meaning,
meaning it produces a lot of stable acids. It uses a process called mixed acid fermentation to break down glucose.
so E. coli loves to make that broth acidic.
You could say that.
Yeah.
Now let's compare that with Enterobacter aerogenes.
Okay.
It's another bacterium that's often found in the human gut. But if you did a methyl red test on Enterobacter aerogenes, you wouldn't get that bright red color change.
So it's not a big acid producer.
Right. It doesn't produce those same levels of stable acids, although it can still ferment glucose just through a different pathway.
So it's like E. coli and Enterobacter aerogenes are both snacking on glucose, but what happens after that snack is totally different.
A great way to put it. And this isn't just a fun fact. Knowing the difference in the ways they metabolize, it actually has some very important implications.
I bet. That's what I love about these deep dives. We start with something seemingly simple, like a color-changing test. But it always leads to a bigger conversation about how these bacteria actually affect us in the real world.
Exactly. And that's really where the methyl red test becomes really important. It might be a simple test, but it tells us so much. Maybe we should spend some time digging into those real-world implications. What do you think?
Definitely. Let's connect the dots between that red color change and why it matters for us. Definitely. Let's connect the dots between that red color change and why it matters for us.
Okay, well, let's think about it. In healthcare, say you need to identify the exact bacteria causing an infection. Right.
That's super important for deciding what treatment to use. The methyl red test, simple as it is, can be really helpful for that.
Because different treatments work better on different types of bacteria.
Exactly. Like knowing whether to use a broad-spectrum antibiotic or something more targeted. But it's not just medicine. The methyl red test is important in food science, too.
Oh, yeah. To make sure our food is safe.
Exactly. Like, think about that yogurt we were talking about. The methyl red test probably helps make sure it's safe to eat and that it tastes good.
Wow, so these tiny organisms and their acid production really affect us in some pretty big ways.
Right. And don't forget about research. The methyl red test is a really important tool for microbiologists all over the place. It helps them learn more about how bacteria work and how they affect, well, pretty much everything.
It just goes to show that the tiniest things can have a huge impact. We've learned so much just by looking at a color change in a test tube. Absolutely. And it makes you realize there's always more to learn. Like we've talked about bacteria that produce acid, but what about the opposite? What about bacteria that neutralize acid or even make things more basic or alkaline?
It's like a whole other world of microbial activity.
Yeah.
So much to think about.
Right. There's always another question, always something else to explore.
Well said. And that wraps up our deep dive into the fascinating world of the methyl red test. It really makes you appreciate the power of observation and asking why.
It really does.
Thank you so much for joining us, and until next time, stay curious.